Hello, everyone. Thank you for the opportunity to address this conference. I'll be speaking on somatic mosaicism, mostly focusing on clonal hematopoiesis and their contribution to cardiometabolic disease. So, we all recognize as an introduction here the traditional cardiovascular risk factors: hyperlipidemia, hypertension, diabetes, smoking, and of course, inheritance. But what we all recognize as well is that the major risk factor for cardiovascular disease, as well as many other diseases, is of course advanced age. And that is not only, as I mentioned, cardiovascular disease and stroke disease, of course, Alzheimer's disease, respiratory disease, diabetes, even cancer increases exponentially with age. So what is it about the aging process that is contributing so much to these types of disease processes? And it kind of gets into the question of why do we undergo biological aging, even at a fundamental level? And what we've become interested in my laboratory is this condition that's referred to as somatic mosaicism, which is basically the time-dependent accumulation of somatic mutations within all the cells in our body. And basically, somatic mutations linearly increase with age in all tissues. It's estimated by age 50, humans will have accumulated more than 1,000 somatic mutations per cell. And this happens in the skin, in lungs, in liver, bladder, brain, of course, the cardiovascular system. And what we will really be focusing on is what's happening in the hematopoietic system. And what's happened in the last few years is that there have been an explosion of manuscripts on looking at somatic mosaicism within human tissues because of advances in technology that allow us to sequence DNA with such precision now that we can see the appearance of these somatic mutations in some cases within individual cells. Now, what are the effects of these somatic mutations on cell fate? Well, of course, greater than 99.9% of these things, these somatic mutations, are neutral mutations. They are just, having no effect on gene function. They have no effect on cell phenotype. Occasionally, sometimes these mutations will hit a vital gene and will sort of inactivate that essential gene, and that will then make that cell give that cell a disadvantage such that it will be removed from the population, and that can be sort of referred to as clonal depletion. However, and probably even more rarely, so these mutations will occur in what's referred to as a driver gene. A driver gene is basically defined as being a gene that provides a selective advantage to a cell, such that it undergoes greater self-renewal or proliferation, or can survive stress better, something like that. And that will give that cell then a selective advantage, and it will undergo a clonal expansion. And the clonal expansions are what are of particular interest these days, because the driver gene will then undergo this expansion as well, this mutated driver gene, and will amplify the mutant's observable phenotype. Furthermore, there's also a pragmatic view of this as well. These are the ones that are the easiest to see. You can see a clone that's basically growing by doing this ultra deep sequencing on human tissues. Now, when this occurs in the hematopoietic system, this condition is referred to as clonal hematopoiesis, okay? And typically, clonal hematopoiesis is attributed to driver genes that are recurrently mutated in blood cancers. And so these are genes such as DNMT3A, TET2, ASXL1, P53, maybe PPM1D, et cetera, et cetera. And what you can see here is that though these mutations are actually occurring in these hematopoietic stem cells, this clonal expansion then occurs over the lifetime of an individual. And so if you're looking at, the relatively large clones that are sort of defined as clonal hematopoiesis, you can see that young individuals really rarely exhibit these clones, but you can see that as an individual's age increases, one can actually sample the blood and do this deep type of error-corrected type of sequencing typically, and see that these clones are basically expanding in these individuals. And I diagram this here below, basically in early youth, these hematopoietic stem cell mutations in driver genes are relatively rare, and the clones are very, very small. By middle age, I like to say that everybody has popped a mutation. If you do deep error-corrected sequencing, you could find that just about everybody has a clone, except they're really very small. But what happens is that under conditions of advanced age, these clones will grow in size in some individuals and lead to this condition that's referred to as clonal hematopoiesis, or CHIP, which is clonal hematopoiesis of indeterminate potential, where 30% or more of the white blood cells in an individual could be derived from a mutant clone. It's really quite striking that these clonal expansions will happen to this extent. And it took a lot of people by surprise when this was realized. So in 2014, there were two papers back to back in the New England Journal of Medicine that really contributed significantly to our understanding of the importance of clonal hematopoiesis in the population. And basically what these studies did was they went in and they were looking for clonal expansions in the blood of individuals that were relatively normal. They did not have a hematologic malignancy. And they found that individuals that had these relatively large clones, because they could only look at large clones at the time because of the technology that they applied, what they found was that individuals that had really overt clonal hematopoiesis had a reduced lifespan. There was about a 40% increase in mortality over the follow-up period, which was really quite striking. And so the question is, well, what was it that these people then developed that led to this death? What was the death? What could you associate this with? What types of morbidity could you associate this with to explain this mortality? And you would think, well, maybe hematologic cancer, because clonal hematopoiesis is basically a precancerous state. So the whole concept here is that, you know, you've already acquired one driver gene mutation. And so this individual would probably be more likely to develop a second driver mutation and a third driver mutation, et cetera, and then develop a hematologic malignancy. And in fact, they did find that there was a striking increase in the frequency of hematologic cancer in individuals that had this precancerous state. However, it turns out that individuals that develop hematological cancer are relatively rare in the population compared to the incidence of clonal hematopoiesis. Whereas the incidence of hematologic cancer is about 30,000 individuals per year, millions and millions of individuals in the United States actually have clonal hematopoiesis. And so this blood cancer risk cannot account for this large increase in mortality. And what was really very striking was the Ji's Wallpaper here. And this was actually just presented in the data supplement. But these, in a secondary analysis, they basically found that probably what was leading to this mortality, the cause of this mortality, were big risks, big increases in the risk of coronary heart disease and stroke. So that provided, you know, sort of the first evidence, epidemiological evidence, to suggest that this precancerous state, or these somatic mutations in the hematopoietic system, could be a risk factor of cardiovascular disease that could maybe be, you know, a sort of a new mechanism of cardiovascular disease that could be adding to the traditional risks of hyperlipidemia, hypertension, diabetes, smoking, and heritable loci that I mentioned before. And in fact, since that time in 2014, there have been a large number of epidemiological studies that have associated clonal hematopoiesis with increases in mortality, as well as many studies that have associated clonal hematopoiesis with an increased risk for cardiovascular disease. I would say that this is pretty much established epidemiology. And just before I move on, I just want to point out that it's not just cardiovascular disease. There have been published associations between clonal hematopoiesis and multiple disease processes, such as chronic kidney disease, solid tumors, colitis. It actually turns out that individuals living with HIV have an increased incidence of clonal hematopoiesis. There's issues of directionality here. It's actually been associated with premature menopause in women. So there's really a lot of pathology that's been basically associated with clonal hematopoiesis. All right. And it's going to be a very rich area both in terms of epidemiology, but also understanding mechanistically what's going on here and understanding the directionality of some of these associations as well is going to be an area of interest going into the future. But of course, we all realize that all this epidemiological work just provides associations. There's really no evidence of causality. It could be that, for example, as we age, we develop clonal hematopoiesis. As we age, we also develop cardiovascular disease, and they're just merely epiphenomena of the aging process. We can't jump to the conclusion that there's a causal connection, that aging then leads to clonal hematopoiesis, and that clonal hematopoiesis is causally upstream from cardiovascular disease. Likewise, we can't say anything about directionality, and that's an area of interest right now in terms of clonal hematopoiesis as well. It could be that as we age, we develop cardiovascular disease, and this cardiovascular disease could be promoting the clonal hematopoiesis. And so really one of the ways that one can start to dissect these questions is by basically building model systems. And so I'm going to tell you some of the work that's gone on in my laboratory where we've looked at some of these clinical associations using mouse models. We've provided evidence for causality. And then through these mechanistic analyses, I think have provided therapeutic insights, and some clinical validation now has come out of the clinic that provided evidence for some of these clinical evidence for what we find in the mice, sort of a clinical validation. So one of the things I want to point out, I don't have a lot of time to speak about this, but we spend a lot of time trying to build bona fide models of clonal hematopoiesis. We've spent a lot of time on this. There are some of these method papers that we've published that if you're interested in this, you could look at some of these papers. But we've really tried to, instead of looking at mice that have hematologic malignancies or sort of other types of hematological problems, we are conducting bone marrow transplantation methodology to look at this, which really better models the condition of clonal hematopoiesis. And what do I mean by that? Well, it's sort of shown here. We will do a competitive type of a bone marrow transplantation where we will actually find that the mutant cell will undergo a clonal expansion, but it does so at the expense of the wild type cell. So there's no change in total leukocyte content, okay? And that is consistent with the clinical paradigm of clonal hematopoiesis. One cannot assess clonal hematopoiesis by doing a blood cell count on individuals. One actually has to sequence the DNA in the blood to see that there's this clonal expansion, because, because these cells are not transformed, all right? They are still, they adhere to sort of the rules of replication, and they do not undergo this uncontrolled proliferation that's normally associated with a cancer, okay? And so when one does that, builds these careful mouse models and then looks at this in models of atherosclerosis. So for example, in an LDL receptor deficient mouse, one can see that there's an increase in atherosclerotic plaque size. And so that provided the first evidence for causality, suggesting that this precancerous state, TET2-mediated clonal hematopoiesis, could be accelerating atherosclerotic cardiovascular disease. Additional evidence for causality, we also looked at models of heart failure to sort of to corroborate and expand our original findings. And we would look at models of both of ischemic on the left and non-ischemic heart failure on the right, whether it's an LAD ligation or transverse aortic constriction. I don't mean to explain all these data to you here, but basically we found that regardless of whether we were looking at ischemic or non-ischemic heart failure, that the condition of clonal hematopoiesis led to worsening of the heart failure characteristics in these mouse models. And we actually published that study in JACC in 2018, a well-regarded clinical journal. Remarkably, they published this mouse study. And I have to say that this mouse study was published before there was any clinical evidence that associated clonal hematopoiesis with worse prognosis and heart failure. And in fact, that clinical validation subsequently was provided by the Zaire laboratory in Frankfurt in this paper by Dorsheimer et al. In JAMA Cardiology in 2019. So in effect, I like to look at this and say that the mouse work was then subsequently validated by the clinical work that was done by my colleague Andrea Zaire. And so, you know, this sort of summarizes this whole thing. We're at the midway point of my 30-minute talk here, and it's a good time to sort of stand back and summarize these things before we move on. So as we age, just at random, we will acquire somatic mutations. Sometimes these somatic mutations will hit a driver gene, such as TET2 in the hematopoietic system. When that happens, these cells go rogue. They undergo this clonal expansion, giving rise to this condition that's referred to as clonal hematopoiesis. And they will exacerbate, based on these animal models, atherosclerotic cardiovascular disease or promote the heart failure pathological phenotypes. So the question came down to, what's the mechanism? And if we understand the mechanism, would this give us new insights into new cardiovascular disease therapies? And there are a lot of possibilities, a lot of possible mechanisms that could explain this. It could be an effect on cholesterol levels, et cetera, et cetera. But what we found, and if you go into our science paper and you look at the supplemental data, there's a lot of negative data in there. The positive data was, we found that TET2 clonal hematopoiesis was promoting pathology by markedly upregulating this inflammatory cytokine IL-1 beta. And we could demonstrate that in cell culture experiments, where we could find that the TET2 deficient macrophages were more likely to produce IL-1 beta when stimulated with various types of stimuli. And we could also look in these atherosclerotic plaques or in these heart failure models, and we could demonstrate that there was more IL-1 beta transcript, there was more IL-1 beta positive protein positivity in these atherosclerotic plaques as well. And the critical experiment was, we then treated these mice models, whether it was heart failure or atherosclerosis. Only atherosclerosis is shown here. We treated these mice with an inflammasome inhibitor that blocks the production of IL-1 beta, and essentially we could reverse the pathological phenotype that was being conferred by the TET2 mediated clonal hematopoiesis. And so based on this, we propose that therapies that inhibit IL-1 beta signaling may more efficiently reduce cardiovascular disease risk among individuals with TET2 mediated clonal hematopoiesis. And then that brings me to the Cantos Trial. Cantos Trial, as you may remember, they were looking at 10,000 high-risk individuals. And they basically were treating these individuals. It was a placebo-controlled dose escalation study. They were treating these individuals with canakinumab, which is a neutralizing antibody to IL-1 beta. They basically found that these high-risk individuals responded to the canakinumab therapy, and canakinumab was shown to reduce major adverse cardiovascular events in the total population that they examined. It actually turned out to be statistically significant at the middle dose, where there was about a 15% reduction in major adverse cardiovascular events in the entire cohort. And if you combine the three doses, if you pool all that data together, there was about an 8% reduction in major adverse cardiovascular events. But when Novartis went back, and this was a study that was conducted by Eric Svensson and his colleagues at Novartis, and they went back and they retrospectively analyzed clonal hematopoiesis in these individuals, they found that carriers of TET2 clonal hematopoiesis displayed a 64% reduction in major adverse cardiovascular events. So as I mentioned earlier, this provided clinical validation of the experimental work that we had done in mice. It also illustrates the clinical utility of measuring clonal hematopoiesis in the patient population. It may tell you how individuals may respond to an immune modulatory drug. And frankly, I think that this provides really compelling information to suggest that that measuring clonal hematopoiesis may provide a realistic pathway for doing precision medicine for the treatment of cardiovascular disease, which is something that people talk about a lot. There's a lot of talk, but there's no action, and maybe this is something that could be actionable. And one of the things I want to point out before I move on to the last third of my talk here is that we've now looked at multiple driver genes in these careful animal models that we construct. And whether we're looking at TET2 or any of the 5 other driver genes that we've looked at, they all seem to function relatively similarly in that they all turn on pro-inflammatory phenotypes. I mean, there are distinctions. They all behave in their own particular ways in terms of the inflammatory cytokines that they dial up. Some are stronger and some are weaker than others. But generally speaking, these mutant immune cells, it predominantly affects myeloid cells. There's a lot of IL-1 beta and IL-6, as well as other cytokines that are being upregulated, sometimes IL-18, et cetera. And they contribute to this chronic inflammatory state. And it probably is one of these things that actually contributes to the chronic inflammatory state that develops in aging organisms, including humans, which has been referred to as inflammaging. But there's an issue here, and that's what I want to talk about in the last 10 minutes of my talk here, is that these clonal hematopoiesis driver genes that are recurrently mutated in cancer, these are the CHIP genes that everybody talks about, things like DNMT3A and TET2 and ASXL1, et cetera. They only account for a very small portion of the clonal events in blood. Individuals can go in and use sort of these non-biased analyses of clonal events, by looking at whole genome sequences or looking at the amplification of passenger genes and whatever, and find that remarkably, there's a lot of dark matter here. We do not know what's controlling a lot of the clonal events in blood. So the question comes down to what other age-dependent mechanisms of genome instability might account for these missing clonal events. And so that's why we became interested in sort of the sex differences here, in terms of mortality and morbidity. I think that part of the rationale, part of the sort of the dark matter here has something to do with this question of why is it that females live longer than males. Life expectancy in the United States at birth for 2021 is that women will live 5 to 6 years longer than men. They live 79 years, whereas men live on average 73 years. And that's not just uniquely humans. One sees that in multiple mammalian species, that the female generally lives longer than the male. And what is the rationale for some of that? And it could be that there's a particular type of genome instability in the hematopoietic system of males that one does not observe in females. And it turns out that men lose their Y chromosome, and it's really remarkably prevalent. In fact, it's the most common human postzygotic mutation in the blood in the human population. If you look at this Nature paper here, which looked at the UK Biobank, if you look at individuals that are by 70 years of age, 45% of those individuals have lost an appreciable amount of their Y chromosome, a variant allele fraction of about 10%. And so if you kind of compare the mosaic loss of Y in men compared to the frequency at which we see CHIP driver gene mutations in the blood, you could see that there is quite a bit more loss of Y than what's occurring, the genome instability, which can be attributed to the clonal hematopoiesis genes. And the thing that's interesting about this is that men that have accelerated loss of Y, they've been able to document, Lars Forsberg showed this, an increase in all-cause mortality, an increase in cancer mortality, and an increase in Alzheimer's disease diagnosis. And Lars has been quoted as saying, I'm quite certain that the loss of the Y chromosome with age can explain a very large proportion of the increase in mortality in men compared to women. But again, this is just an association. It could be that loss of Y is merely just a biomarker of genome instability that happens as we age. And in fact, some of the papers, including, I think, this Nature 2019 paper by Thompson, was sort of providing evidence for that alternative theory. And so what we wanted to do was we wanted to test whether loss of Y would causally contribute to mortality and morbidity. And if so, what's the mechanism? And so when we moved to Virginia a little over 4 years ago, I asked 3 fellows in my laboratory, Doctors Ogawa, Sano, and Horitani, to basically look at the loss of Y problem. They also subsequently received significant help from another fellow, Dr. Shavkin, and we also reached out and collaborated with Lars Forsberg on this, and it led to this paper that we published just a couple of months ago that pointed out that hematopoietic loss of Y leads to cardiac in the blood system, of course, leads to cardiac fibrosis and heart failure mortality. So to address this, and I'll go through this quickly, I want to point out that the Y chromosome is sort of a funny little chromosome. It's referred to as a genetic wasteland and a gene desert. And it's really has prevalent intrachromosomal repeat sequences. But we took advantage of those repeat sequences, and we basically built a lentivirus that would be guided to the repeat sequences, specifically in the Y chromosome. And we basically then targeted the Y chromosome. And you could see that we could then very efficiently eliminate the Y chromosome in these mice that were treated with this CRISPR reagent. And we could even do a karyotype analysis. Here you can see the Y chromosome, and here it's missing in the loss of Y model. We then took these cells where we ablated the Y chromosome and we transplanted them into male mice that had been irradiated to basically create a model of mosaic loss of Y. And when we did this, the results were really quite striking and showed parallels what one finds with men that have accelerated loss of the Y chromosome. That the male mice that have this loss of Y showed a diminished survival, an increase in mortality. They showed a reduction in their cognitive ability. They showed cognitive decline here, both in a Y maze and novel object test. They showed pulmonary disease. They showed renal disease that tended to be fibrotic. And of significance to us, because we look at cardiovascular disease, these animals also exhibited an advanced age-related cardiomyopathy and fibrosis. You can see that as animals age, there's a decrease in their fractional shortening, and this was actually accelerated in the loss of Y mice. It was associated with more fibrosis, more fibroblasts in the heart, and actually it was also associated with diastolic failure as well. When we saw that, we then approached Lars Forsberg and we said, but there's not really good extensive epidemiological data on the on loss of Y with mortality associated with cardiovascular disease. And so he then went and he looked at the UK Biobank, and using the most recent release of the UK Biobank, because it's a relatively young population of individuals and there are very relatively few that have died of cardiovascular disease, but using the latest release where there was about 11.5 years of follow-up, he was able to demonstrate that there is an association between diseases of the circulatory system and loss of Y that was actually dose dependent. So in other words, men that had lost a large proportion of their Y chromosome, upwards of 80%, were more likely to be at greater risk of cardiovascular disease. And you could see that this loss of Y was associated with a subset of cardiovascular diseases that's a sort of consistent with non-ischemic heart failure, such as hypertensive heart disease, heart failure, congestive heart failure, et cetera. And so to move this along faster, rather than waiting 18 months for these mice to develop these types of pathologies, we started looking at an acute model of cardiac overload, transverse aortic constriction. And when we did that, we found also that these mice displayed accelerated decreases in cardiac contractility. They became very fibrotic. The thing that we noticed that was really very weird to us at the time was we did not see any signature of cytokines, chemokines, or inflammatory signaling. And that is so different from what we saw with TET2, which what I shared with you previously, where here you can see the knockout hematopoietic cells basically are really very robust in terms of being stimulated and basically expressing a lot of these cytokines and chemokines. And when we did this bulk RNA sequencing, which is what's shown here, we actually saw what we thought was an increase in the pattern of TGF binding and TGF beta signaling. And so, it kind of is inconsistent with the paradigm where injury leads to inflammation, and then reactive to that inflammation is this increase in fibrosis, which then sort of feeds back and shuts off the inflammation. It seemed that loss of Y was just activating fibrosis independently of inflammation. So to do this, to analyze this at a higher resolution, we did some single cell analysis, and the data was really quite striking. We were able to, in these Seurat plots here, we were able to find macrophages that were either inflammatory or fibrotic, based on the co-expression of markers. And what was really quite surprising to us was that under the conditions of loss of Y, there was a reduction in the inflammatory macrophages and a compensatory sort of increase in the number of fibrotic macrophages under the loss of Y condition. So if you look at this fate analysis, loss of Y is driving these cells towards live 1 positivity associated with fibrosis and kind of an M2 phenotype, whereas clonal hematopoiesis drives you more towards the pro-inflammatory. So it's an anti-inflammatory versus pro-inflammatory. So really quite striking differences here. And just to sort of wrap this up, I've got 1 minute left, maybe even less than that. I'll go very fast. If you look at regulons, by taking this single cell analysis, the single cell data, and looking at loss of Y, the regulons are all being upregulated that activate TGF beta signaling. And if you look at the regulons that regulate IL-1 beta signaling, they're all being downregulated. Based on these data and the data that I showed you in the previous slides, we then treated these mice with a TGF beta neutralizing antibody in the TAC model. And we were actually able to show that when we treated these mice with TGF beta, this increase in fibrotic area and fibroblast number, it returned back down to sort of the baseline. That's what's found in basically in the control mice here. And that was actually associated with the recovery of the contractility in these mice. So in other words, the TGF beta neutralization refers to the phenotype. So just to finish this up really quickly, I just need another few more seconds here. Organ fibrosis, it's a big deal. It increases with age, and it's contributing to many of the deaths in industrialized countries. More than half of the male-specific mortality can be associated with loss of the Y chromosome. It is significant. We think that, you know, it is not only a feature of genome instability, but it's actually, we think it is causal for some of these diseases. We think it's causal because it leads to this dysregulation of TGF beta signaling and fibrotic responses. And as a result of that, we think that men with loss of Y may exhibit a superior response to anti-fibrotic therapies. Just want to point out that loss of Y and CHIP are mechanistically highly divergent. It's just really very, very striking to us. And so where we are now is we can ask a number of other questions. Is loss of Y involved of other diseases and biological aging phenotypes? What are the consequences of LOI and CHIP, loss of Y and CHIP? We look in some of our clinical data, we see that men have both loss of Y and clonal hematopoiesis. What are the loci on the Y chromosome that confer the LOI phenotype, and what are the mechanistic details? And thank you for your attention. I wish I could be with you in Seoul. It's a beautiful city. I've been there a number of times as well. I have many good friends there. Maybe next time. But anyway, thank you for your attention, and I will end this talk now.